reproduction was for several years. Started with our 2016 as a advisory biologist here at University, supervised by myself and Dr. Ed Baker. To be an honor, and during the time, Doug has just done a bang-up job running operations there. We've been employing for the tenure that Doug's been with. Probably 70 people that have worked with Doug every year, including over a dozen graduates. And so we've had the fortune to have a number of persons to work with. With government, what will be associated with
Thank you very much, Kim. Hopefully everybody can hear me. I'm seeing that it's hard to hear. I'm sorry. I'll speak up as loud as I can, everybody. We had some technical difficulties. So I want to start today by thanking my committee. Fortunately, Dr. Ed Baker is not here today from Michigan Department of Natural Resources, but he's also a member of my committee. Sorry you couldn't be here today, Ed. As Kim mentioned, I'm going to be talking about my thesis work, which looks into investigations into reproductive effort. I'm really interested in the idea of current versus reproduction under the guise of life-history theory. That was what I'm doing now. That's what I could do later in life, and what trends are associated with increasing age and interrepair species. So I'm going to focus on that today. Getting started right off the bat, not many people know me in this department because, as Kim mentioned, I've not really been on campus most of the time. I did spend one semester on campus taking a class with Brian and living in my camper. But for the most part, I've been kind of a recluse from campus. Kim mentioned that I've worked at the Black Lake facility since 2016. In my free time, if you want to call it that, I also serve on the XCOM board for Michigan FS. I rotate off in 1 week, so knocking a lot of things out here in the next week or so. I also do a lot of duck hunting. I have 2 wonderful kids. I'm 35, so I'm probably one of the older graduates. 2 wonderful kids, Henry and Madeline, who are 11, 5 respectively, who are watching. And I also serve as an elected official, because when I say free time, I really mean do extra work. So that's a little bit about me. I'm going to focus, like I mentioned, on reproductive value and trade-offs in current. And so in general, the trade-offs between costs associated with resource investment and current year reproduction incurred in terms of future survival and reproduction is what mediates reproductive behavior in long-lived species. So I'll be talking a little bit about reproductive value and reproductive effort and the balance between the two. And they're visualized well on this graph in that we have a 3-axis graph which shows us current fecundity, which is a measure of current investment in reproduction, and then residual value. Effort versus value in reproduction, in which residual reproductive value decreases with age, less reproductive opportunities to reproduce. And current fecundity is a measure of reproductive effort in the current year. And so as you can see here, as reproductive value declines, in this case with age, so does the increase in fecundity. And so what you're looking at here in interrepair species is this constant trade-off between current and future reproduction. And an optimal reproductive tactic is one which maximizes an individual's reproductive value at every life stage. Reproductive effort should vary, as predicted by Jorgensen et al. 2016, should vary inversely with current reproductive value or expectation of future reproduction. And so what I'm interested in lake sturgeon is based on some work that we did in 2020 that we published in the Canadian Journal of Fisheries that looked at how in the current year organisms have trade-offs, or lake sturgeon have trade-offs in that current versus future reproduction. And so what we looked at in that paper is how as river residence time increases, how sperm quality decreases or sperm concentration decreases, and subsequently what that means for current versus future reproduction within a year. This study looks at how that balance between current and future reproduction interplays across multiple years. So as I mentioned, I work with lake sturgeon. Lake sturgeon are a species of conservation concern, threatened or critically endangered in 19 of the 20 states in their home range. And the combination of habitat degradation, overharvest, river impounding, all contributed to the beginning decline in lake sturgeon populations in the 19th century. To the point now where lake sturgeon populations are estimated to be at about 1% of their historical maximum. And restoration efforts are ongoing. So I work with the Black Lake facility. It's an excellent example, I think, of a restoration effort that's ongoing in the Great Lakes, but other sturgeon restoration efforts are ongoing throughout the Great Lakes region. A couple of interesting features that make lake sturgeon such an interesting study in the context of life history. They're long-lived. Sturgeon are theorized to live over 100 years, and they exhibit delayed maturation. So lake sturgeon males can take up to 15 years before they're reproductively mature. Females closer to 25 years. They're patadromous, which means they invest heavily in migrating from a resource-rich lake environment into a riverine environment, and that is particularly costly. So that's one of the things that's I think really interesting about lake sturgeon and current reproductive trade-offs. They're highly fecund. Over the course of a lifetime, a female lake sturgeon can carry between 4,000 and 7,000 pounds of eggs, and in each reproductive bout, they can be expected to release somewhere between 300,000 and 500,000 eggs. And they're interopaired, so they spawn multiple times, multiple reproductive bouts, which is particularly interesting. And then one thing to really note here is they are highly periodic in their spawning. And so male lake sturgeon, at least in Black Lake, can take between 1 and 2 years to reproduce, whereas females can take between 3 and 5 years to reproduce. And that's the basis for one of my chapters today, is investigating how that interspawning interval can vary over the course of a lifetime. And they're broadcast spawners, which means they spawn in large groups. Operational sex ratios in the Black Lake are about 2.5 males to every female during the reproductive period. And they reproduce throughout different areas of riverine systems, and we'll talk a little bit more about that in a moment. Lake sturgeon also exhibit spawning site fidelity, which means at least in our system, we can expect to see females return to the same approximate location to spawn annually. And the standardized date of spawning is highly repeatable. And what I mean by that is relative to the first day of when a female in particular can be expected to spawn is highly repeatable. And so we like to view that as early, mid, and late spawners, or early and late spawners in our system. And so you can expect a female that's always an early, or the female that's an early spawner to always be an early spawner, and vice versa. River entry, and this is important, river entry is influenced by the lag effect of discharge. So that period of roughly 72 hours, sturgeon start migrating is important. We see a decrease in discharge, an increase in temperature coupled with lunar phase, generally brings sturgeon into their migratory period. And then spawning success is highly dependent on a number of things. So in particular, females are heavily and availability of mates. So that leads me into the first chapter of my thesis, which is lake sturgeon interspawning interval is associated with prior reproductive effort, including intra and intersexual behavior in response to environmental conditions. So as I mentioned, in 2020, we published a paper in the Canadian Journal of Fisheries and Aquatic Sciences that looked at a single year of reproductive effort for lake sturgeon and how behavior could vary within that season. And we found that lake sturgeon behavior in a spawning season, at least in this system, is highly variable. Some males will reside in the river for long durations during the spawning period. Others will migrate multiple times in the season to avoid costly river retention. But the net result was that whenever we saw, we quantified the increased number of females, number of available mates available to a male, an increase in female mates ultimately led to an increase in reproductive success. In addition, longer river residence time was associated with increased female encounters as well. So that combination of increased residence time, more female encounters. However, this did present a within-year trade-off where males that stayed in the river. So it's a really interesting study of how 1 year can result in a trade-off between current reproductive success and the possibility of depleted resources. And so one other thing that's really interesting about lake sturgeon is they exhibit dimension spawning previously 3 to 5 years for females. And that's not something that's unique to sturgeon. We see that this figure demonstrates how interspawning interval varies for a number of fish species. It varies as a function of. So x-axis, you're looking at longevity. Y-axis, you're looking at non-reproductive periods. I've highlighted two fish species here. White sucker and lake trout both have varying degrees of longevity, where white sucker are expected to live into the range of 10 years, whereas lake trout upwards of 40 or 45 years. And what you'll notice of fish species that are that have a shorter longevity is they also have a much shorter or much less percentage of their life cycle spent in that context. So we expect to see white suckers enter reproductive periods early in life, and the time that they spend in non-reproductive periods is pretty short, meaning they don't generally sit spawning out. They're available. Contrast that with lake trout, which are much more longer lived, and as a result, spend roughly half of their life, indicating that they too exhibit some skip spawning. And so this is really interesting because the question I'm asking is what leads to the skip spawning, or what things are associated with skip spawning in lake sturgeon in particular. So lake sturgeon are interesting, and we've been able to, because we have this population monitored, we've been able to evaluate inner spawning interval over multiple years. And so what we've been able to demonstrate is that for males, we do see some 1 year period of spawning, and that about 41% of the spawns in year 1 can be expected to return in year 2. The remaining 59% or so that spawn in year 1 can be expected to return in year 3, or in this case, time 1 and 2. What that's really pointing at is that about 41% of the population in a given year returns to spawn in the successive year. And roughly the remaining 59% or so will come back in that successive 2-year period. Quite a bit different for females. As I mentioned, the periodicity is quite a bit longer, and that a female that spawns in time T, population-wide, none of that population, nobody. In addition, we expect to see roughly 6% return after 2 years. Additional 57% return after 3 years. And my mouse froze. There we go. 25% after 4 years, and the remaining 11% will come back after 5. So we're looking at average spawning periodicity of about 3.19 years for females and 1.61 years for males. So with that in mind, the objective of my first experiment was to document whether or not any behavioral, biotic, or environmental variables were associated with variability in inner spawning interval for both male and female lake sturgeon, and whether or not inner spawning interval was associated with changes in environmental conditions during the spawning season. So I have two main hypotheses from this chapter. The first is that inner spawning interval will increase as a negative function of body size. So I would expect that young fish will have a longer inner spawning interval compared to older fish, which I expect to have a much shorter inner spawning interval. That's predicted by life history theory. The second hypothesis that I have for this experiment is that year-to-year differences in inner spawning interval are associated with variation in both spawning behavior and environmental conditions. And so that's what I'm investigating in chapter 1. So rather than showing you a long list of things that we do to fish and how we capture fish, because this is such a unique system, I always like to take an opportunity whenever I can to show this video. This is how we collect lake sturgeon in the upper Black River. So as you can see, I'm wearing a GoPro in this video, and we dive for lake sturgeon. And when we encounter them, we simply scoop them up in large full-bodied nets, and we return them to the surface, to the shore, where we can do full workup. I'll note that the Black Lake system is relatively shallow and narrow, and as a result, we're able to do we're able to collect about 70% of the adult population. I'll also note that this system is impounded both on the inlet and outlet, so I'm dealing with a population that features very little emigration or immigration. Once we get the fish to the surface, we bring them up on shore, where we measure biological variables, including fork length, total length, girth, weight, and the presence of gametes. And we also tag fish, including with radio frequency identification. It's focused on data collected from radio frequency identification. We do that with 23 and 32 millimeter Oregon RFID pit tags. And because this system is narrow and shallow, we're actually able to build these systems out ourselves. The system itself is essentially a passive pit tag reader that covers the entire duration of the spawning period of the spawning surface in the known spawning area. And as I mentioned, about 95% of this population at this point is tagged with radio frequency identification tags. As I mentioned, I'm working in the upper Black River. This study takes place between 2016 and 2022, and we use RFID antennas throughout the entire stretch of the river, including one antenna at the beginning of the known spawning area, a second set of antenna at the beginning. The first antenna is at the beginning of the mouth. The second set of antennas is at the beginning of the spawning area. We have one antenna upstream of the known spawning area, and then we have another antenna all the way up at Kleber Dam, which represents the uppermost limit, which can dispersed in the system. And in some years, particularly high water years, we do see fish as high up as Kleber Dam. Using radio frequency identification allows us to calculate a number of variables based on when we capture and when we capture. For example, we're able to collect data on upstream swimming time, the number of times in a year a fish migrates the full system. So that's defined as an entry to the system, entry to the spawning grounds, and an exit from the system. And we count the number of times that happens in a season. We're also able to monitor river residence time, or the time between a fish entering the river and exiting the river, assuming they make it all the way up to the spawning grounds. And collecting these variables allows us to essentially get a surrogate understanding of the behavior that fish are exhibiting in the system. River residency in particular is a really good surrogate for effort expended to remain in the river. We also use hobo loggers throughout the system to collect a number of environmental variables, including total discharge, total critical thermal units, both measures of conditions in the river. We also calculated this variable that we call days in the river above 5% exceedance flow, which essentially represents all of the flows that exceed 5% of the historical max. So there was a gauge installed in the Upper Black River right below Kleber Dam from 1942 to 2000. We queried all of the results from that gauge and found the value at which 5% value was exceeded. So the maximum discharge in the system was 52.67 cubic meters per second. And the value of the upper 5% of all discharge values was about 16 meters per second for the system. And so what we're looking to quantify there is high flow days. So any day in which the river flow or discharge, excuse me, is about 16 meters per second is in the upper 5% of all flows. Historically or colloquially, the upper 5% of all flows is consistent with a 20-year flood event. In addition, we used the generalized linear mix model, which was Poisson distributed, and accounted for random variation that could be associated with multiple capture events. In this case, we used RFID for that. We also used a dummy variable observation to help us deal with some underdispersion in our data. And we used year as a dummy variable to try to see if we could explain any variation just based on year. And we used these random variables to see if we were getting any contrast with the random variation that was in the sample versus the fixed effects that we measured. And so the first step in doing this was to take the fully parameterized fixed effects model and look at the different possible combinations of random variables to determine whether or not random variation in this sample could be explained by one of these random variables or a combination of these random variables, or if it was simply best explained by fixed effects. And then because of the differences that are associated with male and female reproductive effort, we analyzed males and females separately in the study. The first thing I want to talk about is how river residency time varied across years. And so some of these environmental conditions we know are associated with river residence time. So we wanted to evaluate across the study how river residence time varied. And I want to draw your attention very specifically to 2018 and 2019 in this graph, particularly for males. Note that there's a pretty sizable difference in river residence time here, where males in 2018 were expected or were measured to be in the river for about 172 hours on average, compared to 2019, where males were in the river for about 283 hours. So a pretty substantial difference in time spent in the river during this period. Same for females. 2018 was the year in which female river residence time was lowest, whereas 2019 was the year in which female. And in general, the variation in river residence time was between about 119 hours for females overall and 199 in the highest year. And one thing I want to point out here is one thing that we know from previous research is that increased river residence time is associated certainly with increased reproductive success, but also increased river reproductive expenditures. We also evaluated how fish were exposed to cumulative discharge during their period. And so cumulative discharge is a measure of all discharge experience while the fish was in the river. And we noted a few things here. First, cumulative discharge experience is different between sexes. Just like river residence time, males and females are behaving differently as a result of environmental conditions during the spawning period. What we noted here was that the lowest overall discharge experienced in the river by fish was experienced in 2016, where fish were exposed to about 47.32 cubic meters of discharge per second across the time that they were in the river. In addition, females were also experiencing their lowest overall discharge measures in 2000. So 2016 in general was a low discharge year for us. Compare that with 2020, in which we actually measured the highest overall discharge ever recorded in the Upper Black River at about 53 cubic meters per second. Overall, we saw higher cumulative discharge in that year. And what I want to note too here is, again, cumulative discharge is relatively similar between 2018 and 2019. River residence, we'll touch on that here in just a minute. Finally, looking at those extreme temperature day or extreme flow days, those 20-year flood event days, pretty similar overall findings to what we found with cumulative discharge. The number of 5% days was significantly different across years and between the sexes. And we noted that in 2022, basically none of the fish experienced flood event days compared with 2020, where we had our highest overall discharge. And some fish experience, on average, male fish experience is up to 7.5 days in the river in which we were over flood stage. So there is a ton of variation in this sample in these high discharge days, just like there is a ton of variation in overall discharge between years. But again, I'm going to note here, 2018 for males in particular, you can see not a ton of difference in cumulative discharge or 5% flow day. So 2018 experienced pretty big differences in river residence time, but didn't really experience huge differences in environmental conditions. And so the question that begs is, what does that mean for the duration and morphology of the spawning? What does this mean? Why am I seeing higher residence times in some years when environmental conditions are relatively stable? And what can I expect to see? So I'm going to highlight 4 years for you here: 2018, 2019, 2020. In 2018, you'll note discharge was extremely high at the beginning of the spawning run. And as a consequence of that, the spawning run started quite a bit later in 2018 than it did. In fact, the duration of spawning in 2018 was also quite a bit lower. We experienced 35 days of total spawning in 2018, which represents the lowest overall duration of spawning during the study period. In addition, in 2019, we experienced the longest overall duration of spawning period. You'll also notice in this graph that there's very little variation in discharge and temperature during the spawning period. So as I've already addressed, that 72-hour lag effect of discharge dropping and temperature increasing can cue fish into the river. But relatively stagnant environmental conditions after those cues can also keep fish in the river. And what we saw in 2019 was that fish stayed in the river for 57 days. In 2018. Additionally, you'll notice differences in the morphology of the spawning. In 2018, because the run was so compressed, we really had one pretty much continuous period of spawning during the season, with a one-day kind of separation, if you will, runs 1 and 2. In 2019, we saw 4 peaks of spawning throughout the season, and very little wasted spawning periods, in that when males were in the river, so were females. There's very good match between males and females during this year. They were in the river consistently with each other for the entire duration of the spawning period, and as a result, river residence time for both males and females was highest in 2019. Contrast that with 2020, again, highest overall discharge year, but that discharge dropped off quite a bit earlier and stayed low for the duration of the spawning. So while we did see a second peak in discharge resulting in clear dual modality of the spawning period, what we ultimately were left with was what we would consider to be a relatively reasonable spawning period at 55 days. So not that short duration like we experienced in 2018. Finally, I'll highlight 2021 because this is an interesting case study, the possible complications of climate change going forward, in that we had a long duration of spawning. Fish were in the river this year for over 57 days. However, there was a very dramatic match and mismatch between when females and males were in the river this year, because we didn't experience that dropped pulse and discharge or increase in temperature. Females came in consistent with the lunar phase, but males didn't. Males took a full 18 days to come into the river in any significant quantity after females were already in the river. You'll note that there were only about 18 days of good overlap between males and females spawning in 2021, and then males were in the river on their own for another 10 days. So even though we had a long duration of spawning this year and river residence times were generally high, the trade-off was that because of that late season increase in temperature, females left the river and there really wasn't good spawning periodicity this year. And again, dual modality spawning. So collectively, what these results demonstrate is that there are clear differences in environmental conditions between years, and these changes in environmental conditions can result in changes in river residence time, which is important because we know river residence time is associated with reproductive success. And then one other thing I want to highlight here is the changes in interspawning interval or population-wide probability of spawning as a result of some of these differences in environmental conditions. So the first thing I'll note is that when you look at female periodicity, so explaining this graph, I guess I should do first. What you have here is initial period of spawning, year +1, year +2, year +3, year +4, and year +5 on this axis. And this corresponds to initial year of spawning of 2016, 17, 18, and 19. And diagonally, what you're looking at here is the resulting periodicity of spawning. So for example, this value here of about 0.527 is consistent with males, which initially spawned in 2016, and what the periodicity or what proportion of the population came back in year +1, whereas this value of 0.38 is consistent with years of fish that came back in years +2. So what we find here is that there's really not a huge difference in what we expect females to do between. Not only there were some differences, but not significantly different between years. So those females that spawned in 2016, 2017, and 2018, there wasn't a huge change in the probability of return spawning in the successive years K1 through K5. And that the entirety of the fish that spawned in 16, 17, and 18, nothing came back after year 1. In 2016, about 11.8% of the population came back in the second year, but down to 7% from those fish that spawned in 2017. Overall, though, no significant difference between these values between years. Males, on the other hand, experienced pretty sizable shifts in proportional return in successive years as a function of year. For example, in 2016, where I've already mentioned the discharge was really low, and subsequently river residence time was moderate to high, the population returned at about 52.7% in the successive year. So male fish that spawned in 2016, about 52% of those fish came back in 2017 to spawn. Same thing for 2018. About 52.4% of the population that spawned in 2018 came back the next year, compared to about 42%, which came back in 2 years. So in 2016 and 2018, we're seeing shorter residence times, and as a result, potentially higher proportions of the population that could come back in the next year. Interesting example of a trade-off between current and future reproduction. 2018, really high discharge, really short duration of spawning. As a result, a large amount of the population is able to come back in the next year. Contrast that, for example, with 2019, where the majority of the population was in the river for a very long time. River residence time was very high. Discharge was very low. Fish invested very heavily in that year of reproduction. And as a result, only 34% could come back in a successive year. So some interesting variability in interspawning proportion as a function of those environmental conditions. I'll note that this population is 1,183 individuals, and we did this over a capture of over 1,738 captures in 2,614 RFID detection events. I'll note that size, and this is important for the rest of this chapter, size is very variable between the sexes in this population. And the average female is about 150 centimeters at breeding age in this population, compared to males, which are an average of about 126 centimeters in breeding age. And so we do see some differences in the proportion of differences in the fork length of the population. That's really interesting because it leads to the question, what does that mean in terms of interspawning interval? If females have a longer interspawning interval, they're larger at spawning, does that mean the same thing for males? So we evaluated that, as I mentioned, using a generalized linear mixed model. What I was looking to do here first is determine whether or not random variation in the sample could explain what was going on with interspawning interval before I tried to look at fixed effects. So I used that fully parameterized fixed effects model in order to evaluate whether or not random variation in the sample could explain what was going on with lake sturgeon interspawning. And I used Akiaki's information criterion to evaluate these different combinations of random variables, along with the random variable free model, to determine for males and females if there were any random effects that should be considered. And the first thing I want to note here is that no, there are no random effects that need to be considered in the sample. The best model for both males and females is one which did not include random effects. So I can be confident that the fixed effects that I use explain variation better than random effects associated with individual return, with observational period during the season, and differences between years. So what I wanted to establish is whether or not if there were any variables that were fixed effects that were behavioral, biological, or environmental that could explain why female interspawning interval varied across the population. And essentially what I found here is that there were no fixed effects that we measured that were associated with female interspawning interval. None of the fixed effects that we were going after in this survey, whether it be time spent in the river, behavior in the river, or the environmental conditions in the river, none of these were associated with female interspawning. Males, on the other hand, we were able to explain with variables that we measured in the sample. In particular, we found negative associations between interspawning interval of male lake sturgeon and intersex interactions and fork length. So what these two variables are looking at here are surrogates of reproductive expenditure. Whereas on the left of this graph here, we have young males, and And on the right side of the graph, we have these older and larger males. And what we see is that as these lake sturgeon are aging, as they get older, the spawning periodicity is decreasing. And so it's what we would expect to see as predicted by life history theory. Older males that are more robust and have future residual reproductive opportunities seem to be spending less time between spawning periods and are instead investing in making sure to have multiple or to have more current reproductivity than, for example, these smaller males that are not entering the river as often and have that longer spawning periodicity. So female interactions, as female interactions increase, so also our inner spawning interval. And that's another way of saying that these fish that are invested really heavy in this current reproductive bout are likely older, more invested fish with fewer reproductive opportunities available. As a result, they're seeking to increase their number of female encounters and they're reducing spawning periodicity. So lake sturgeon body size, at least in males, seems to be associated with a decrease in inner spawning interval, which is something that we would predict as under the theory of life history. To round off this chapter, conclusions from this chapter, males, there was a strong negative association between body size and intersex interactions, which indicates an interesting trade-off between current and future reproduction. And from a theoretical perspective, the decline of inner spawning interval with age and body size is consistent with what we would expect to see as predicted by life history theory, which predicts that the spawning interval should be the longest immediately following first maturation and should decrease with age. And this has been seen in other interapparent spawning species, including Atlantic cod and lake trout. In addition, female inner spawning interval doesn't seem to be associated with anything we measured here, and I offered two explanations for that. One is the long periodicity of female lake sturgeon means that we only really had the majority of our recapture events, we were only one-time recapture, right? So we captured those fish initially, we captured them when they returned to the river to calculate inner spawning interval, and we did not get many successive capture opportunities with those females. Contrast that with males, which have a much shorter spawning periodicity, and were theoretically possibly caught in every year of the study. And in fact, we did have multiple males that we caught every single year of this 6-year study. So it could be that we just simply need more data on females in order to really be able to parse out these fixed effects. Alternatively, female inner spawning interval could be strictly resource-based. So lake sturgeon spend like a long period of time in the river or in the lake between spawning periods. They're capital breeding species, which means resource acquisition is super important in order to be able to get into a reproductive state. It's much more energetically costly to produce eggs than it is sperm. So it could be possible that female inner spawning interval is simply associated with that time needed for resource acquisition, which we didn't evaluate in this study. Finally, the seasonal reproductive period is quite variable between years. There's evidence that we see a plastic response to that in decreased population-wide inner spawning probability. And in general, we do see some evidence that perhaps climate change can provide us with an opportunity to have some match and mismatch between males and females in that environmental cues may change as a function of how inner spawning, or excuse me, how climate change may affect, for example, early spring warm up or lower snowpack leading to lower discharges during the winter months. So with that, I'm going to move on to chapter 2. Focused on inner spawning interval and life history theory. Chapter 2 focuses on cryptic female choice and the possibility that ovarian fluid may mediate selection between closely related and unrelated males. So gamete fertilization success in broadcast spawning fish is largely mediated by properties of sperm, including sperm quality, in this case quantity, excuse me, concentration and volume, and quality, i.e. velocity and duration of motility. In some species, moderation of sperm motility by ovarian fluid may provide some degree of cryptic female choice for or against males of different phenotypes or against relatedness, for example. So I present two examples of that on this slide. The first, as presented in Alonso et al. 2016, demonstrates the relationship when ovarian fluid is not present between sperm concentration and probability of fertilization. So if you take a male, in this case, a nesting male, which is the preferred male phenotype in oscillated race, which were studied by this paper, when ovarian fluid was not present, as sperm volume increased, so did proportional overall fertilization. When ovarian fluid isn't present and you increase the amount of sperm available, reproduction increases well for that. However, when ovarian fluid was present in oscillated race, we see a pretty consistent proportional fertilization by the nesting male strategy. And that's because ovarian fluid seems to equalize that sperm volume advantage in oscillated race. In addition, Gasparini and Flastro studied Guppy, and they found that ovarian fluid may mediate sperm velocity, particularly when compared between an unrelated and a related male. They used half siblings in the study, and they found that when ovarian fluid was present on a related egg, sperm velocity of that related male was decreased relative to the unrelated. This increase in sperm velocity, because sperm is so important to reproductive success, could theoretically translate to more progeny being produced by that unrelated male, thereby allowing the female to cryptically select against the related phenotype. In addition, Gasparini et al. Really demonstrated this idea of fertilization success as a function of ovarian fluid being present, as they did a competitive fertilization between this unrelated and related male, and found that the unrelated male does produce more progeny when ovarian fluid is present. So there is a theoretical mechanism for cryptic female choice demonstrated by Gasparini et al. Here. So my question, and this begs the question, do broadcast spawning species which exhibit really limited pre-spawning mate selection and post-zygotic parental care, do they have some sort of mechanism for post-gamete release selection? And this is important because captive breeding programs in particular have relied really heavily on wild egg and sperm collection in order to produce progeny. So my objective for this study was to determine whether ovarian fluid was a medium by which female lake sturgeon could mediate cryptic choice by affecting fertilization success or egg survival by males, and to quantify whether or not sperm quantity or quality was affected by the presence or absence of ovarian fluid on eggs. I did this across 3 experiments, in which one was a fertilization experiment and other was a computer-assisted sperm analysis experiment. My hypotheses for this experiment were that proportional fertilization and survival of eggs fertilized by half-sibling males would be lower overall when ovarian fluid was present on the egg surface. And for my computer-assisted sperm trial, my second hypothesis was the presence of ovarian fluid from a half-sibling male will reduce sperm quality and measures of sperm velocity as evaluated by computer-assisted sperm analysis. I did this across 3 experiments. In order to do this, we used the genotypes of 1,320 individuals that have been genotyped in this system since 2000, to generate all possible male and female relatedness combinations for the system. The entire Black Lake population has been genotyped at 13 disomic microsatellite loci. And we used the program ML-Relate in order to determine relationships between a female and multiple males in the system. The program ML-Relate was used to calculate a maximum likelihood estimate of pairwise individual relatedness and to discriminate against general pedagogical categories, including parent offspring with a relatedness value of 0.5, full siblings with a relatedness value of 0.5, half siblings with a relatedness value of 0.25, and unrelated individuals with a relatedness value of 0. As I mentioned, we did this across 3 experiments. In the first experiment, we looked at how sperm volume could affect fertilizations in general, so that in the subsequent experiments, we weren't over or under fertilizing as a result of sperm concentration or quality. We did a fertilization experiment in which we competed males that were unrelated to a female versus a male that was related to a female. And finally, we looked at that sperm under a microscope when ovarian fluid was present and absent to determine if there were differences in sperm quality and sperm velocity. One interesting finding from this population, because we have such a large and robust genetic data set, we were able to determine that of the 1,742,400 possible male and female pairings, the most common dyadic relationship was unrelated, with about 79% of the population possible combinations being unrelated. So about 17% of the possible combinations of half sibling, about 3% of the possible combinations being full sibling, and finally about 2.1% being representative of parents and offspring relationship. To conduct the first experiment, the sperm volume experiment, we used 1 male across 3 females and 6 reps of each treatment variable. Here we were looking to evaluate whether or not a number of different sperm volumes would affect overall proportional survival or fertilization success. So sperm volume is our treatment variable. Our dependent variables were proportional fertilization after 24 hours and proportional survival at 48 and 72 hours. In order to evaluate this, we used a generalized linear mixed model with a beta distribution that evaluated proportion, and we accounted for the random effect of female parentage and the effect of multiple sampling periods. And very simply, what we found in our sperm volume experiment was that when we encompassed the range of sperm volumes reported in the literature in combination with the possible sperm concentrations reported in the literature in 2020, this range of sperms really had no effect on proportional fertilization survival, proportional fertilization or proportional survival after 28, 48, and 24, 48, and 72 hours respectively. In other words, none of the 5 volumes that we used really had any effect on the probability of reproduction or of successful fertilization in this experiment. And from this, we were able to then decide that we could default to our existing standard operating procedures, which would allow us to use 20 microliters of sperm, which was one of those sperm concentrations reported. With that in mind, we moved on to experiment 2, in which we evaluated whether or not the presence of ovarian fluid on an egg when a male is related to or unrelated to a female would result in a reduction in proportional fertilization at 24 hours or a reduction of proportional survival at 48 or 72 hours. Here we have 2 males, an unrelated and a half-sibling male, fertilized with 4 females across 6 reps. We had 2 treatment variables, in this case, related versus unrelated, and rinsed versus unrinsed. And then, of course, our dependent variables were proportional fertilization and survival. Again, we used a generalized linear mixed model with the beta distribution and accounted for both the random effect of female parentage and sampling period. Excuse me. The results of this experiment were pretty interesting. First, I'll note that at 24 hours, there was no difference in fertilization success across the 4 different combinations of treatments. So unrelated males in which ovarian fluid was present or absent, and related males when ovarian fluid was present and absent. The result is that we did not see any reduction in fertilization success after 24 hours. So we really didn't see that evidence of ovarian fluid mediating fertilization between related and unrelated males. What we did see, though, was after 72 hours, a notable 17% drop in proportional survival in the half-sibling treatments, and this was regardless of whether or not ovarian fluid was present and absent. So there does seem to be some sort of a deleterious effect after 72 hours on these eggs. However, we can't associate that with ovarian fluid, and we can back that up statistically by noting that the two-way interaction between ovarian fluid and relatedness was not significant. Were it significant, that would suggest that there was a higher effect of ovarian fluid on proportional fertilization and survival as a function of relatedness. We didn't see that interaction be significant. All we saw was differences in relatedness and time in this. So it provides us some evidence that perhaps ovarian fluid isn't being used as this selective medium as we hypothesized. So with that in mind, we wanted to confirm these results looking at sperm concentration and sperm quality and velocity using computer-assisted sperm analysis. So in the final experiment of this chapter, again, we used 6 replicates across 2 males and 4 females. These are the same females that we used in the first experiment so that we could directly compare sperm velocity and quality to fertilization success. And we had 2 treatments here, unrelated versus half-sibling pairings, and then whether or not sperm was activated in an assay of just water at 200 microliters or a combined assay of ovarian fluid and river water at 100 microliters apiece. The dependent variables for this analysis were a composite measure of sperm quality, which we calculated with principal components analysis, and 3 different measures of velocity, including velocity straight line, velocity curvilinear, and velocity average path. As I mentioned, we used principal components analysis in order to generate sperm quality, and that's a method that we developed in the Larson et al. 2020 paper. And we did account in this paper using general linearized mixed modeling for the random effect of female parentage. So what I'd like to see in these experiments is, again, that two-way interaction between ovarian fluid and relatedness, suggesting that differences in velocity and sperm quality are a function of ovarian fluid presence, absence, and relatedness. So the first thing we did was create this composite measure of sperm quality. And I won't spend a lot of time on this, but essentially what you're looking at here is 2 significant axes which explain more than 85% of the variability in our sample. Principal component 1, which is the principal component we were able to use for this study, is associated positively with measures of sperm quality and linearity, sperm velocity and linearity. So velocity average path, velocity straight line, and linearity were all positively associated. We took the factor loadings from that analysis and we used those as our dependent variable in our analysis. And the short answer to what we found was that adding ovarian fluid does seem to reduce principal component 1 on that axis. However, it doesn't seem to reduce the ovarian fluid principal component as a function of relatedness. So in this analysis, what you're looking at here is that the half-sibling was already higher on principal component 1 by about 0.98 units, and ovarian fluid presence does reduce principal component 1, but that principal component 1 doesn't change as a function of whether or not the fish was related to the male or female. So in short, what we would expect to see here, if there was a mechanism for slowing sperm velocity more in the half-sibling versus the unrelated male, is that we would see this two-way interaction between ovarian fluid and both significant and negative. We're not seeing that here. Indicates that sperm velocity does, principal component 1 does drop as a function of the presence of ovarian fluid, but doesn't seem to be affected by relatedness to the female. And this trend continues throughout all of our analyses where we look at just specific measures of velocity. Again, adding ovarian fluid reduces straight line velocity by about 40.57 micrometers per second, but that's pretty consistent between both the related and unrelated male, in that there was no significant two-way interaction. So that reduction in velocity on is a function of ovarian fluid being present but not related. So again, no significant two-way interaction. Same results in velocity curvilinear. We see a reduction of about 48.23 micrometers per second when ovarian fluid is present, but that doesn't discriminate based on. So we're again not seeing that significant two-way interaction. Then finally, adding ovarian fluid to the assay did reduce velocity average path by about 43.8 micrometers per second. But again, that didn't discriminate based on relatedness or unrelatedness of the male to the female. So no significant two-way interaction again. So what we're looking at here in conclusion is that we do see some deleterious effect in proportional survival after 72 hours, where we see about a 17% drop in half-sibling proportional egg survival after 72 hours. But this lack of significant two-way interaction between the presence of ovarian fluid and relatedness suggests that ovarian fluid isn't a medium for cryptic female choice, at least in lake sturgeon females here. In addition, we also see that we have a reduction in principal component 1, sperm quality, and measures of velocity when ovarian fluid is present, but that reduction is not related to whether or not the male is related to a female. And this is important because following release, a possible explanation for this is that following release, male gametes from freshwater fishes have a really short lifespan, due in part to the differences in osmolarities and extra and intercellular fluids to changes in potassium. And so the result is that when an egg or sperm is released into the water, we see pretty quick reduction and decline in sperm viability and egg viability as a function of contact with water. And so a paper from 2021 by Cotolini et al. Offers that in acetopensiforms, rather than acting as a mechanism of choice, ovarian fluid instead acts as an ion buffer, which increases the overall longevity of both egg and sperm survival when they come into contact. And important to note here, if egg viability and sperm viability increases with time, it also corresponds with an increase in the number of possible mating opportunities as both sperm and egg increase longevity. This is also particularly important, I think, from a management perspective, because of course, as I mentioned before, managers usually treat ovarian fluid as an afterthought, especially in selective breeding programs. And we generally don't have the tools necessary to evaluate relatedness in real time. And so being able to understand the role of ovarian fluid, I think, is pretty in captive breeding programs. So that summarizes chapter 2. And with that, yeah, that's it for me. I'd like to acknowledge a few people before I finish up today. First, I'd like to acknowledge that funding was provided by the State Wildlife Grant for this project in conjunction with the Michigan Department of Resources, for which we're longtime partners, 20 years as of last year. In addition, I'd like to thank the Black Lake chapter of Sturgeon for Tomorrow for providing funding that was necessary to purchase additional RFID equipment for this experiment. I'd be remiss if I didn't mention the Black Lake Sturgeon Facility cruise from 2016 to 2022. We put these kids through the ringer, particularly during the spawning period, because lake sturgeon don't take reproductive Saturdays and Sundays. They're in the river when they're in the river. And I couldn't have done this without some of these folks. I'm going to highlight very specifically some folks. Jake Kimmel in particular, who's actually in the room with us today, who was a technician on this project for a year and then a graduate student for 3 years, who was instrumental to this project. I'll highlight Max Majinska. Not a lot you can say about Max, except for he's really the hardest working person I've ever met. And he's been on this project now for 5 years and has now gone on to do a graduate project himself. And he's been instrumental. And I'd like to highlight Lydia Wasink, Amber Johnson, and Joe Reedy as well for their constant support during this project. as you can imagine, doing a graduate project while supervising 8 bodies a year can be frustrating. And so to have these folks around to be able to help out has been instrumental. And then lastly, and I think most importantly, I'd like to acknowledge my son, Henry Larson, my daughter, Madeline Larson, and my partner, Marissa Larson. I did a lot of crazy stuff during this project, including living out of my camper for a semester on campus. And through all of it, they were incredibly supportive, including Marissa, who carried the brunt of parenting for 6 months. So huge thanks to them. And then finally, I'll acknowledge my committee, Dr. Ed Baker, Dr. Brian Roth, and Dr. Travis Brennan. And not least, I'll recognize Kim Scribner for just being incredibly amazing to me over the last 6 years. A lot of work that we do at Black River, and it can be intense. And Kim is one of those people that will be there to do that work with you and would never ask you to do anything that he's not willing to do himself. And he provides tons of support and tons of autonomy. He's given me opportunities to do service commitments that I really enjoy. And of course, he took me on in this master's project, despite everything else we had going on. So really, Kim, I can't say thank you enough. I'll never be able to thank you for everything that you've done for me and my family, but I'll at least take this forum to thank you for all of that now. And with that, I'd like to highlight just how long this project took, because that's what I looked like when I started. And I definitely don't look like that anymore. And I'll offer this opportunity for anybody to ask any questions that you may have. Well, we'll start with the room. Is there anybody in the room that has a question? Jake? That's an excellent question, Jake. We didn't, as the short answer. We didn't see any of those females come back in 2022. Actually, 2022, interestingly, was a really high efficiency capture year. We caught something like 85% of the population. And despite the fact that we caught that many, we just still didn't see very many returnees. And it's starting to look like that in some of the preliminary analysis I've done too. It's the same case with males. Not seeing a lot of those males that were in the river in 2021 return at that high clip like we saw in 2018. Any other questions from the room? Yeah, for those of you online, the question was, is this monitoring continuing so that we can continue to collect long-term data for females? Absolutely. In fact, we start in a week, or 2 weeks here. So yeah, we'll be out there, I mean, hopefully in perpetuity. This is a long-term management project. This population is supposed to be supplemented until it gets to a point where we see some signs of natural reproduction. So it also exists as a partnership between a number of private and public institutions. And so there is some interplay with that. But as of right now, through at least 2026, as far as I know, but it should continue in perpetuity. And as I mentioned, we've been ongoing for 20 years. This is actually our 21st year. So I expect to keep collecting this data as long as they'll let me. Yeah, so the question was, in summary, the question was, do we see some of this non-synchronous behavior with interspawning interval in other sturgeon species? And the short answer to your question is yes. In general, females have a longer interbreeding period in other migratory sturgeon species that I've investigated. Yeah. Yes. Yeah. Yes. Yeah. That's so good question. I guess I could have explained this better, so sorry. The question was if in chapter 1, if the random effects model were considered when fixed effects were present. And the short answer to your question is actually those random effects models that we evaluated were fully parameterized fixed effects and random effects. Exactly. Yep. The only thing we varied between those models was the random effects. So there were 8 possible combinations, one in which all the fixed effects were present and none of the random effects were present, and then different combinations of those 3 random effects. So what we ended up picking there, that last model that you saw, the one that was no random effects was the best model, was the no random effects, but fully parameterized fixed effects model. So all the fixed effects models, fixed effects were there, just no random. Well, now I'm going to dwell on it. Yeah. Oh. Yeah. Yeah. In my case, I was simply interested in determining whether or not the random effects added to the model. So I didn't consider that, but now I'm going to. So thanks for the feedback. I already said, Travis, that I'm going to consider that. So that's off limits. I'm kidding. I guess I'm not seeing any more questions in the chat. Oh, I just got one. So Andrew Briggs asked, sorry if I missed this, I had to exit for chapter 1, but did you see any adults return to the river but not spawn? Andrew, it would have been difficult to quantify that. We see fish that come above F05 bridge that come into the river that never make it up to the spawning grounds. And so that we don't include those in our analyses because we have no evidence that they made it up to the spawning grounds. We do see fish in the river that don't have gametes present, but generally that's a product of not being at the spawning period. So I'm not, in my data set anyway, I'm not familiar with any fish that came up in the river but did not attempt to spawn. Yeah, and exactly, Kim added an absence of genetic data. Looks like we have another question here from Brianna. Do you think that herpes virus in this population is going to be another factor to consider going forward with reproduction success? Like, do you think that there is something that will have implications for river residence time? That's an interesting question, Brianna. I would actually, as much as this is a cop-out, I would direct you to Amber Johnson and Tom Locke's lab for that, as I didn't actually evaluate anything to do with lake sturgeon herpes virus for this experiment. But I could, as Kim noted, we could. Another question in the room, Tyler? Yeah. Right. Right. Sure. So for those of you listening online, Tyler asked how our fertilizations in chapter 2 compared to what we'd expect to see kind of in a natural riverine. We'll summarize it in that way. And I guess the first answer to your question, Tyler, is there are some things that are similar, some things that aren't. So we used reported sperm egg combinations that were consistent with what we saw in the literature. So, there are papers that provide estimates of how much sperm can be expected to be extruded in a cloud, and the resultant relationship with how many eggs are released. hard to really quantify that. But there are some other lab papers. Stephen Tucker's paper from 2020 comes to mind, that do present, possible scenarios for fertilization. Now, an obvious difference is that when we did our fertilizations, they weren't competitive in that the unrelated male and the related male weren't fertilized together, right? So that's not going to be something that you'd expect to see in a natural pairing, where you would expect to see essentially a giant glob of fish fertilizing all together all at once. So we were trying to control for that. So that was kind of a univariate type analysis. So in that respect, not necessarily related to a natural fertilization. Conversely, the volumes of sperm that we picked, the volume of eggs that we picked were consistent with other references in the literature that dealt with sturgeon fertilization, and I guess in theory deal with sturgeon fertilization in the wild. I got one more question from Ed Baker. So I probably should answer this one. For fertilization experiments, which do you think most closely mimics natural conditions, rinsed or unrinsed? I think that's a great question, Ed. I think that the evidence suggests that from that Cotolini paper that I mentioned, that ovarian fluid acts as a sperm preservative. In general, I would say that it's probable that ovarian fluid is still present on the egg when sperm comes into contact with the egg, but is pretty quickly rinsed away afterwards. So I guess the natural conditions that it would most closely mimic would likely be unrinsed rather than rinsed, even though these guys are broadcast spawning species that are throwing eggs everywhere. I would think that at least in the initial period, at least from my kind of anecdotal experience, rinsing eggs off of ovarian fluid in the lab, it takes quite a bit to get that ovarian fluid off the egg. So I would expect that the unrinsed conditions are more consistent with natural conditions. All right.
Okay, well, let's thank someone.
Very much appreciate everybody who attended online. I'm going to close the room, but thank you again very much for coming today. I appreciate it.